Hello, and welcome to my 15-minute wrap up of the lectures that are going on in this week. And so we're going to do 15 minutes for this particular lecture, and we're going to cover both Monday and Wednesday's lecture. Again, this is for specifically January 31st, which is the Monday that we had. Basically, what we're going to be doing is covering unit number 5, which is looking at the evolutionary history of biological diversity. And so basically, what organisms are out there and kind of what are they doing? Our first part is that we are going to look at phylogeny, which is basically our branched history. You know, what did our ancestors do? What did their ancestors do? Who were they? What did they look like? And then what this is going to go into is us going through and knowing the organisms, knowing their history, knowing their relationship, knowing their similarities. And, you know, again, part of that is looking at systemics. And so I am going to share my screen. Let's share the screen if we go on here. And so again, for this week, this is week number 3. We're covering chapters 26, 27, and 28. We will have an in-class working assignment on Friday, so please make sure that you bring your textbooks and your materials to work on for that. I will post this area right up there. We had our validation quiz on Wednesday. We could talk about that a little bit. This is a slightly different thing, open note, open books, and I'm glad you took it. The labs that we were doing this week are lab number 2 and lab number 4. Next week, we are going to be doing another set of labs. We're going to be doing a dissection lab, and we will be doing a little bit of a physiology lab. And I will post those materials pretty soon. So to go on to it and kind of really quick, and I only have 15 minutes to go over basically what I was lecturing, or what I would lecture in 3 hours, or normally lecture in 3 hours. In chapter number 26, we're looking at the tree of what we call phylogeny in the tree of life, of that, how are things related to one another. Again, when Linnaeus went over his materials, he had things more like a ladder, and you looked at things, and so basically all the lower level things, you know, the small plants and stuff like that, and slugs and snails were on one line, and they would look at all those materials and name those materials. And so basically what he was doing was cataloging. What we want to do, you know, and what biologists found out that, and we're looking at it, and, you know, and after Darwin came up with this, with his idea of kind of evolution, and Wallace and a number of other people, it just kind of made sense. And that was a really good way to kind of organize things. And we see a lot of times in science, we get a lot of stuff out of being able to organize things. So, you know, the periodic table of elements was a way to organize kind of what's there. And, you know, again, this phylogeny is just a way for us to kind of really look at life and how they are interrelated. Again, when we look at a species, just superficially look at a species, we could see things that look the same. And, you know, and again, there's a picture here of where we have a legless lizard. And so the loss of limbs, the loss of legs to do various things like swim in the ocean, like dig down in burrows, has happened a number of times throughout the evolutionary process. And if we just like the things and put legless things together, we would put certain lizards together with snakes, possibly even together with whales and porpoises, etc. So, so what are we really looking at here? And what I'd like to say is that, actually, let's go on this. And this is a picture from your book, and I'll do the slideshow on the picture in here. But what we want to do is kind of through phylogeny, what we were going to do is to look at lots of the characteristics of organisms, and not just things that are superficial and can relate to different things. But what we're going to do is try and separate out groups by certain characteristics, right? And so again, what we have on the left-hand side is a common ancestor. And here we're looking at vertebrates. So we had basically a common ancestor that started, you know, again, this is after looking at organisms coming together, becoming multicellular, and other activities that are going on. But once we started to have things get to be what we call vertebrates, then if we look at vertebrates, we'd have to conclude everything, and a lot of times we may be, we're just looking at human beings. When we say vertebrates, we have to include everything that has a spinal cord or everything that remotely relates to a spinal cord. And in this case here, we'd be looking at fishes, frogs, humans, geckos, snakes, etc. And this isn't kind of all inclusive on there. We're picking and choosing kind of things. But what we can say is once we get vertebrates, we could do things like what are the four limbs? And again, you can look at fish, they have fins, but they don't have bones in those fins. And again, we can look at the bony fishes that have that. And that's another reason to look at it. But then that would separate out the fishes from the rest of these. And then when we look at that again, the ancestor characteristics to all of these guys here would be, they don't have it in here, but all of these guys here look like they have, different characteristics separated from frogs. And so if you kind of know frogs, you kind of have this kind of wet skin. You have a rudimentary respiratory system, et cetera. So if we look at things that are a little bit more evolved than that, we can have that. Of those that we could look at, we can have ones that have hair. Again, we could look at placenta. There's a whole bunch of other different things that we could look at for this. For this particular one, the separation here would be maybe looking at opposable thumbs that would separate us. So, again, when we look at phylogeny, we may be looking at just kind of one characteristic, so we're looking at several characteristics. But the idea is to really dig deep and look at specifics. And so again, in lab number 1, in our observation lab, the first question I asked you is if an alien came to our planet and would look at human beings, how would we describe human beings? And a lot of you said, we have arms and legs, we have a head. That's the same thing with the chimpanzees. We have hair. We have hair on certain spots of our, that could be chimpanzees. Again, some of the things that separate us from chimpanzees is we have this kind of higher order of thought. And so even though chimpanzees have a society, they don't have it to the extent that we have. They don't build cities. They don't work together. They don't have kindergarten, right, for their baby chimpanzees. And so again, we need to look at the specifics and what's going to separate us. And so if you really stand back and say, look at it, what are these characteristics? What are we looking at? And so again, in a phylogeny, what we're doing is we're trying to pick out certain characteristics that would allow us to tell the difference between certain organisms. And so I'm going to go back here and some of the things that we are looking at. Again, in naming of organisms, I'm not going to go on to kingdom, class, order, etc. I do want you to know that there are 3 kingdoms of life, and we're parsing out kind of what life is and what the parts of life are and what makes one characteristics better than the other. Okay, and so again, when we visualize the tree, and I'm going to go on my slideshow again that we're looking at there. Again, when we look at this tree, you look at the left-hand side, we have a common ancestor. It doesn't mean that you evolved from something. So we're getting away from that ladder look where you had one rung and then you had the next thing kind of come up on top of it. What it's more kind of like is a bush. And so frogs, lizards, chimps, and human beings, if we go back far enough, we have this kind of separate organism that may be well be dead, and that may, and we all may look very different from it now, or we may not look that different from our common ancestors. But again, a common ancestor to fish and human beings is not really going to look like either a fish or a human being for the most part. And then what you need to do is, if you look on here, we'll have an ancestor in that lineage. And so even though we say frogs here, frogs didn't just step out or become a new species from that common ancestors. There's a whole bunch of kind of intermediaries inside of there. And sometimes the change was really slow and sometimes the change was really fast. And so we do look at that. When we look at some of these trees, and I'm going to look at this. So part of this is you need to be able to look at and read a phylogenetic tree and look at phylogenetic relationships. We can use a vertical tree or we can use a diagonal tree that we're looking at this. These materials here can kind of flip around when we look at it. So that's one of the changes that we can kind of look at. And then where do these phylogeny trees? Can we use these? And yes, we can. And so in this case here, this is a study that was done on whales and actually going into the fish shops in Japan and looking at whale meat and seeing what it is. And so what we would look at, and this is, I believe this is looking at mitochondrial DNA, and minke whales from the southern hemisphere are in pretty good shape, stuff like that, and those can be hunted. But what we found here is that these samples were very close to and related to like minke whales from the north, humpback whale, and maybe the finned whales, some of these large finned whales. And so it's very serious, this is very serious because these are organisms that are greatly protected. And so again, looking at the phylogeny is able to help us with that. So what we want to say in the class, the great tool that we have now is looking at DNA, and you'll get a chance to look at DNA, and we'll develop, we actually have a lab a little bit developed for you. So this is greatly simplified, but let's say we have two organisms and we have a sequence in them. And again, when we look at the sequences, sometimes sequences inside of a gene is going to be conserved. So there are certain genes that we actually see in all living things have very much almost the same sequence. So for like things like cytochrome C, you're going to see very little change in that because again, we can't make cytochrome C any kind of better than it really is. However, most of our genome, 85% of our genome used to be kind of what we call junk DNA, but it's really, we just don't know what the use of the DNA is. And sometimes it's just in there as a spacer, sometimes it's doing something else, some kind of regulation. And so when we look at the DNA, we could have things like a deletion where we remove a nucleotide. We could have an insertion where we add some nucleotides inside of there. And so what we do, if we do this in sequence insertion, we get these two sequences and they look really different from one another. However, if we use computers and algorithms and certain rules, we can actually line these up. And as if you can see looking on the bottom, you can see here where we have the CCAAT, a match. We can identify an insertion in there, and then we can identify that deletion. And so this allows us to say that these sequences, even though they look totally different, are actually quite similar and they share some common ancestor. So both of these are different from that original 1 and 2 that we have up there, but we can see that there's a relationship between them. And again, knowing the rules of how DNA switches will allow us to work on that. And so I'm looking at my timer and I am almost up. And so, and phylogenetic trees allows us to do that. Another thing that we were looking at on chapter number 27 is looking at bacteria. Bacteria are what we call the prokaryotes, are very interesting organisms because they reproduce very quickly. They're very simple organisms. We think they're some of the organisms that first developed on this planet. Because again, we have this idea that life we had the kind of molecular evolution, and that led to a living cell. The key features to prokaryotes are that they are very small. They reproduce by binary fission. Basically, they make another copy of themselves and they generate, and they have very short generation time. So these guys can evolve relatively quick because again, there's lots of them that are out there and they reproduce a real lot. So that when the mistakes are made, those mistakes will die off and they'll quickly get replaced by other organisms. When we look at life though, and we're going to see this a lot, we look at a lot of kind of where they get their energy. And so prokaryotes can be categorized according to how they obtain their energy. And we have phototrophs, which obtain their energy from light, chemotrophs, which obtain their energy from chemicals, or possibly even rocks and materials in an environment. Autotrophs require carbon dioxide, and heterotrophs require complex organic molecules. And so they need to have molecules that are fixed by other ones. And again, phototrophs would include things like blue-green algae that are able to grow. And the next, in chapter number 28, we're looking at the protozoans, which you got a chance to look at in chapter number, actually experiment number 3, we're looking at, actually experiment number 2, we're looking at microscopy. And again, the protozoans are small single-celled eukaryotic organisms. And so that's one of the big things. You do need to know the difference between eukaryotic and prokaryotic organisms. These guys are interesting because, again, we have photoautotrophs, which contain chloroplasts, which are able to photosynthesize. We have the heterotrophs, which obtain organic material. And the mixotrophs are some that have a little bit of both. And that would include an organism like the euglena that you heard about. Well, that's it for my kind of 15-minute wrap up. I will be publishing the study guides for the next materials, and so look for that soon. And with that, I'll say have a great day. The sun's coming out, and enjoy yourselves. I'm going to stop my share now. My alarm is going off. Bye now.